Welcome to my tutorial on how to use Beegle for disease gene discovery. Beegle is an online search and discovery engine that starts from literature mining to automatically identify non-disease gene annotations. Then it integrates multiple genomic models to further generate novel hypotheses. Beegle is designed in a Google-like interface in which we can enter our query of interest here. This, for example, can be a disease name or a phenotype or a combination of both. In this tutorial, I would choose Alzheimer's disease as my query, and then I will press search for known genes. The first phase in Beegle presents us with an ordered list of the genes that are found to be linked with our query according to the literature. We can see here that there are 185 genes linked to Alzheimer's disease, and we can easily navigate through this list forwards and backwards. Per gene, we can find some details. For example, we can check the supporting publications in which this gene and our query are mentioned together. If I click here, I can go directly to the list of all publications on PubMed. And if I want to check this specific publications, I can also click on the PubMed ID. This is repeated for every gene in the output list. So, for example, I can here again see the supporting publications. I can also see the word cloud for my gene. For example, for this gene, PESN1, the first representative keyword for this gene is PESN1. I can also see that Alzheimer's disease appears the third representative keyword. I can also compare this word cloud to that of the query by clicking here and also viewing the word cloud of my query. I can see that PESN1 also appears in like the top 10 representative keywords for this query. And finally, I can download the list of output genes by clicking on download known genes, and then I can save it on my local machine. In order to proceed for the next phase, I have to click here, proceed to discovery. To start this phase, I first need to select a training set, which I have three options for. The first option is to select the training set from the search results that Beegle just presented. The second option is to select manually a training set, and the third one is to upload directly a training set. So in this tutorial, I would choose select genes from search results. I can easily select all the search results by clicking here, or if I want to individually select those 5 genes, for example, I can easily do this. If I'm doubtful about any of my training set, I can again check directly the publications and the word cloud for that specific gene. And I again can compare it to that of the word cloud. I can also search for a given gene, which however needs to be present in my known set of genes according to Beegle. So let's say, for example, APP, for example, here, and then it can easily come up even if it's not in the first page. So now I would like to select those 5 genes as my training set. I just press here add selected genes. I can see now my list of training genes. Then the second step is to select my candidate set. The default is to use the whole genome and prioritize the whole genome given this training set. However, the user can also select manually the candidate set of interest. And here, Beegle presents to us the list of all the possible genes that can act as a candidate set. And if I would like to, for example, select a gene that I know by gene symbol, I can simply search for it and select it. Or if I would like to again select another gene, let's say APP. However, this is one is from Alzheimer's, but anyways, I can again also select it, and then now I'm chose my candidate set. I just press add selected genes similarly, and then here I can see my added candidate set. Now, if I would like to edit this set, it's also possible. I can just press here, click here the gene that I want to remove, and just remove the selected gene. And this can be also done for the candidate set. Now I've selected my training genes. I've also selected my candidate genes. The last thing I have to do is just to press discover. Then Beegle automatically sends a request to Endeavor, which does the genomic data fusion in order to prioritize my candidate set. However, this can take some time, like, I don't know, maybe 30 minutes or something. It depends on the size of the candidate set. But for the sake of this tutorial, I would directly show you the output list for another example. I also used Alzheimer's disease as a query. However, I asked Beegle to prioritize the whole genome. So this came up with almost 20,000 genes. The presentation is similar to the search phase, in which I have an ordered list of the genes that are prioritized according to the different genomic sources. Also per gene, I can find some details about it, like, again, the supporting publications and the word cloud of this gene, which I, again, can easily compare to my query. And I can also go back to the training set for this specific run. So for example, in this run, I only chose those 5 genes as a training set. And that's why I can see, for example, APP as the top gene prioritized, but this is because it was not used in the training set. Finally, I can download the list of prioritized genes by clicking this button, similar to the search phase. Now, if I would like to start a new query from the beginning, I can simply select the query here. For example, I would like to choose diabetes, simply diabetes, and I just press search. And again, I'm presented with an ordered list of the genes that are known to be linked with diabetes in a similar way like any other query. Thank you very much for watching, and good luck with your prioritizations on Beegle.